Good morning, everyone. Thank you for joining us. We're going to give everyone another 30 seconds or so to hop on, and then we will get started. All right, everyone. Thank you for joining us. It's now 8:02. We're going to get started today. Here we have Aaron. Aaron is our CSO here at Twist Bioscience. Prior to Twist, he served as the CSO of Lake Pharma, leading the California Antibody Center. He also oversaw all discovery research functions as both the VP of Protein Sciences at Surrozen and as Vice President of Research at Sutro Biopharma. He also held director level positions at both Oncomed and Diax Corp. We're very excited to hear Aaron speak today to Twist Bioscience accelerated antibody discovery workflow, highlighting advanced high throughput characterization assays and the strengths of the DiversimAb platform. We'll do our best to leave time for Q&A at the end of this session, so please send over any questions you might have during the presentation via the QA button you'll see at the bottom of your screen. And with that, I will hand it over to Aaron.
Thanks, Chrissy. Again, my name is Aaron Sato. I'm the CSO of Twist, and today I'm going to be walking through all the amazing things that are happening in our Boston site, which uses the DiversimAb in vivo antibody discovery platform. And I'm going to be, I'll go through the discovery process and how it works, but I'll spend the bulk of the time going through all the advanced characterization tools that the Twist Boston site uses to characterize all of the amazing antibodies that come out of the mouse. So here we go. So the Twist Biopharma Solutions group overall, including the Boston site, which focuses on in vivo antibody discovery, is very focused on using mice as well as alpacas, as I'll talk about later, to discover both fully mouse-derived IgGs and heavy chain only antibodies from alpacas, using both hybridoma for mouse antibody discovery, and then single B cell cloning for mouse antibody discovery and alpaca B cell antibody discovery. The site is industry leading experts in discovering antibodies from in vivo sources, wide range of expertise in both generating antibodies for therapeutics as well as reagent antibodies. And as I mentioned, they're based in the Boston area, which is the largest hub for biotech in the United States. So they're local to many of the companies that we work with in this space. So in terms of the in vivo offering for Twist Biopharma Solutions, it's really focused on these main kind of pillars of functional readouts for the group. So the first one is, of course, designing your antigen, which again, we'll be using for in vivo immunization. Again, that could be oftentimes a protein or a peptide or a cell line that overexpresses the antigen. But again, we also can use DNA or RNA as an immunogen as well to get tighter against the specific target. And then we'll go through a whole advanced series of immunization schemes for generating that immune response in our DiversimAb mouse, which has a broadened immune repertoire, as I'll talk about later. We also can do work with partners that have human antibody transgenic animals as well. So you're not just restricted to using our mouse, which we also think is great, but you also can use other mice as well in concert potentially with the DiversimAb mouse. And then, as I mentioned, we also can do immunization in alpaca to generate heavy chain only or VHH antibodies. On the screening side, once we have titer, we then need to isolate our specific antibodies. And so, as I mentioned, on the mouse side, we can do that through traditional hybridoma. But we really love to use kind of advanced technologies in single B cell cloning, in particular on the Beacon Berkeley Light System, where we can screen, again, thousands and thousands of single B cells that have for unique binding as well as functional activity on the beacon. And we kind of the holy grail for antibody discovery is not only just assessing binding, but also assessing function early on. And that's what the beacon vertically light system allows for. We can actually assess for not only high affinity binding of the antibodies from the single B cell clones, but also whether they have functional activity and maybe one or two specific assays that the customer requests. And as you'll see later, we can get a broad repertoire of sequences from this workflow. And once we have those sequences, we can do some nice assessment of sequence liabilities based on those primary sequences. But the real meat of the presentation I'll talk about today is really about the optimization and characterization of those antibodies. So once we have the sequences, we'll of course make recombinant versions of those mouse antibodies or VHH antibodies from alpacas, and we'll go put them through a whole high throughput series of tests for binding affinity, of course, epitope binning, developability assessment. And once we have our leads, we can then potentially do some additional antibody engineering and optimization, as well as humanization. As I mentioned, the antibodies coming from our DiversimAb mouse are wild-type mouse sequences. They do require humanization, but we have some really advanced tools for doing bulk humanization, which I'll talk about later. And then finally, all of those antibodies can be expressed in multiple formats. We have a high throughput IgG offering in our South San Francisco site using XB293, as well as the Boston site also uses XB293 to make large amounts of recombinant material. And again, we can validate the binding and characterization that we previously did early on in the screen. So just to kind of talk about the types of targets that we can go after using this platform, it really spans the gamut. I mean, I think in general, I mean, a lot of our customers come to us once they've with really challenging targets that they may have actually tried on their own, but have failed. And so they come to the experts to help them with discovering antibodies to targets like single span cell surface targets, multi-span GPCRs and ion channels, glycans, for example, highly homologous targets that might oftentimes be difficult to generate in mice. We've been successful time and time again against multiple targets like this. In addition, as I mentioned a couple slides ago, we're also doing a lot of work in VHH discovery from alpacas, and we've shown that and validated that we can do all that workflow on the Beacon Berkeley Light system. And we're just about to publish, we actually did publish a paper quite recently, which you should check out, showing how we use the Beacon system for VHH discovery. And then finally, we have a lot of partners in the CAR-T, CAR-NK space as well, where we're helping them discover binders as part of their CAR platform. So let's talk about some of the challenges in antibody discovery against some of these really next generation difficult targets. Sometimes it's really difficult to generate the recombinant protein. And so as I talked about before, we can work on generating cell lines or maybe use DNA as the immunogen to maybe get around this. We can work on other ways to engineer the protein for greater expression. And so we're not only experts in antibody discovery, but we're also experts in protein sciences as well to enable us to make these really difficult antigens that are important for our antibody discovery work. And as excuse me. And as the mantra we always say is, garbage in, garbage out. We spend a lot of time making sure that the input antigens are at the highest quality so that we get high quality antibodies out of our process. Another thing that's, again, oftentimes a challenge is poor immunogenicity and getting that relevant titer. So again, as I mentioned back a couple other slides, the DiversimAb mouse itself has a very open immune system, which allows it to generate a more robust immune response oftentimes compared to a lot of other wild-type mice, or maybe even some of the transgenic mice, even for really similar targets that are have similar human mouse, even cyno cross-reactivity. And then finally, even though sometimes we use recombinant proteins, we're big fans of doing cell surface screening early on, because oftentimes, as you know, as we all know, that sometimes we can generate antibodies that bind the recombinant protein, but don't bind the membrane-bound version of the same protein. So we can incorporate that in the beacon screen potentially, but also once we get to crude soups, we also can very quickly do high throughput FACS screening as well, which I'll show a little bit later. So getting back to the advantages of the hyperimmune mouse, which I tout all the time. I think it's an amazing mouse, and it's not the only advantage of using this overall platform, but it's a big part of it in the sense that it allows you to generate antibodies to a broad range of epitopes that the standard mouse might not, just because oftentimes with standard mouse immunization, you're really only generating antibodies to the sites on the protein that are different between the human protein and the mouse protein based on tolerance. But the DiversimAb mouse has a much more open tolerance system. And so you can actually generate antibodies to epitopes that you oftentimes wouldn't be able to do in a standard mouse system. So that's a huge advantage of the system. And in comparison to many of the other wild-type mice, or even some of the transgenic mice, we can get much broader epitope coverage than many of those other mouse systems. And this slide just kind of brings that message home, that in this specific example against target X, we were able to get over 53 human cyno cross-reactive clones. Of course, we've got several human specific clones as well, but mostly cross-reactive clones. And then we're also able to get a lot of human mouse, even cyno cross-reactive clones, which is awesome because, again, as we go into both preclinical studies as well as IND-enabling cynotox studies, having an antibody that cross-reacts with all three is really ideal. But oftentimes in mouse immunization systems, it's really difficult to do. But the DiversimAb mouse, hyperimmune mouse, lends itself to that. So, and this is an example on the right showing the broad epitope coverage you can see for this particular antigen compared to the BALB-C comparator in yellow. For the DiversimAb mouse, we did both a rapid as well as an extended immunization. And you can see, if you just focus on the purple and orange colors, you can see that you get a lot more epitope bins for DiversimAb relative to BALB-C. So again, bringing home the message that much more open immune system, much less more tolerant to other similar sequences between mouse and human, and that gives us greater shots on goal against our target. This slide goes through our single B cell workflow, as I mentioned, for mouse. I mean, we can do either single B cell cloning or hybridoma for mouse, so we have a choice. But we really love the single B cell cloning platform because it enables us to screen so much more and get so much more sequences out for your campaign. So we really strongly suggest our customers use our single B cell cloning approach. And again, it starts with similar workflow I talked about before, antigen projection, generation and immunization to generate that repertoire, throwing that on the beacon system to screen out your single B cells. And again, as we do that, we can do very extensive a number of different assays, not just binding assays, but also additional functional assays for which I'll show later to assess whether your antibody has specific functional activity, as well as potentially even looking at species cross-reactivity. So really amazing system that allows you to get a lot of data really early up front before you select your clones to propagate further and actually test. Once we have those sequences, as I mentioned in previous slides, we can do all kinds of different additional tests for sequence, cell binding, sequence, species cross-reactivity, blocking, non-blocking, as well as kinetics and epitope binding, as I'll show in subsequent slides. And then finally, once we have our top mouse hits, in the case of coming out of the DiversimAb hyperimmune mouse, we need to humanize them. And as I'll show, we actually have an amazing informatics team as well at Twist, where we can do bulk humanization, and then because we're a DNA company, we can rapidly synthesize them, make the antibodies, and do all the downstream tests. And those tests will include all the testing I talked about before, but also a whole host of developability tests, which I'll spend some time talking about, because again, developability is just almost as important as binding affinity and function in terms of taking antibodies to the clinic. And as you will see, because we get so much diversity out of our mouse, we can really cherry pick the best clones that have the right affinity, the right specificity, as well as great developability. This just goes into more detail about the Beacon Berkeley Light System. I'm sure a lot of us have seen demonstrations from Berkeley Lights, but also from potentially from us as well. Again, it's an amazing system where you can pen and isolate up to 14,000 single B cells per chip. We could, once we do an immunization, we of course can do multiple chips for each campaign to expand the diversity of sequences that we potentially can get. We can do, as I mentioned, sequential assays to identify and provide that data to pick specific hits. And then once we have, and we know which pens have the single B cells that we like, we can export those for single cell sequencing. And then we can, once we have those sequences, we can, of course, do the recombinant production and all the downstream testing that I'll talk about today. This just gives you a flavor for all the different assays that we can run on the Beacon. So as I mentioned, as we talked about before, we can do a whole host of cell binding assays, which may be actually very important to do if it's like, for example, a GPCR target, a multipass transmembrane target, ion channel target. You won't have like a recombinant protein version that you can put on a bead in the Beacon. So here cell binding is really important, and you might not even just want to do a cell binding test early on to assess that difference between recombinant protein binding and cell binding. Oftentimes we're coming up with antibodies that are blocking for either the ligand or the receptor. We can actually run receptor ligand competition assays on the Beacon to give us an assessment of whether we actually potentially have antagonists of that specific interaction. We, of course, can do multicolor assays to look at, say, for example, species cross-reactivity between mouse-human cyno, as I talked about before, or highly homologous proteins that we want to test for binding or non-binding to within a specific family. And then we even can do like really custom functional assays as well, looking at, for example, internalization, apoptosis, as well as other fluorescent readouts that you might want to do. So as I mentioned before, the team in Boston is industry leading experts in using the Beacon system, and it's really these kind of more complex assays that are really fantastic to use as part of your campaign. And it really goes to show that you can get so much data early on in the process rather than later on in the process to really help you pick out the best candidates to move forward. Okay, so this goes into that bulk humanization I talked about before. So once we have our mirroring antibody sequences or our alpaca sequences, as I talked about before, we can put them into a whole series of databases that we have internally at Twist. And that allows us to rapidly humanize them using multiple algorithms to generate humanized sequences. And then once we have those versions of humanized sequences, maybe multiple versions for each specific mouse or alpaca sequence, we can, of course, rapidly synthesize them and then make the resulting antibodies and look for comparability in terms of affinity and function relative to the mouse or alpaca version of the specific antibody. And all of this work is royalty-free and doesn't add any additional cost in terms of downstream economics using this approach. And this slide just kind of goes through that in more detail in terms of the bulk humanization offering, in terms of the in silico piece of it, which is very quick. And then the specific synthesis and testing and reconfirmation of binding. And then, of course, we'll talk later about how we can add on a whole series of developability tests as well to assess not just comparability in terms of binding and function relative to the mouse counterpart, but also do we also achieve similar or maybe better developability as we humanize as well. So another great strength of the in vivo team in Boston is their downstream characterization ability, which I think is, again, second to none in the industry. So we can, of course, do high throughput or medium throughput expression purification of the antibodies. So as I kind of breezed over it in the last slide, we can do up to, on the high throughput side, we typically do about a 5 mil transient transfection and HEC-293 to give maybe about upwards of 1 milligram of antibody for doing a lot of these tests. And then another thing that we can also do once we have our leads is we also can scale them up as well to give larger amounts of material. And we have automation X as well as purification systems to allow us to make and characterize those antibodies on HPLC. In terms of the downstream characterization, of course, we can do ELISAs to assess specificity and binding of the antibodies. But as you'll see in the subsequent slides, we have a lot of expertise and SPR and BLI to assess both affinity as well as epitope mins, which is second to none in the industry. And as you'll see as well, we can use crude soups as we do that to really enable and speed up the process so we don't have to go through purification. Of course, as we talked about before, doing flow cytometry is also really important, and we have several IQ screeners to enable us to do high throughput flow cytometry to look at binding to the immunized target versus the background cell line, and then maybe even some other transiently transfected targets that are either homologous that you don't want binding to, or maybe other species of the target that you also want to assess on cells. And then finally, we have a whole host of developability assays, which we spent a lot of time validating to look at from SEC to CIC to HIC, AC-SINs, PVP ELISA, as well as thermal stability and temperature of aggregation. So all these great parameters will, again, allow us to narrow down our functional hits that we really like and really focus on those that are highly developable going into the clinic potentially. Okay, so let's move this over here. So in terms of doing high throughput cell-based assays, we work really closely with our partners to really assess exactly what they want in terms of the downstream characterization. This is one example where we worked on a cell surface receptor. It contained 9 highly conserved, there was 9 highly conserved members in the family. And the partner was looking for a therapeutic antibody. They wanted to be specific to that one family member without any off-target binding. They wanted functional ligand blocking activity was required. They wanted a diverse set of sequences, of course, and they're on a tight timeline. So it sounds really familiar. And again, we went through the process I talked about before, from immunization, harvest, plasma B cells, going on to the beacon, sequence, and then of course we only moved forward those hits that met all these criteria. We of course sequenced them, expressed them, and then put them through a whole panel of functional validation to see if we've met all of these key criteria. And shown here is just a snapshot of a lot of that data. So shown here on the bottom left is a nice correlation between ELISA, the antibody resulting antibodies, and the MFIs on flow cytometry, which again is a nice good indicator that antibody that binds protein is also binding to the resulting target on cells, which is great. And that's probably because we probably immunize with the cell surface receptor on cells, or maybe potentially with DNA. Shown here is just MFIs looking at multiple different other family members to assess, the level of specificity against family member 1 versus the other family members. And this gives you a nice pictorial view of a couple of those clones that show really nice specificity to FM1 over the other family members. And then at the end of the day, we expressed 96 clones that came off the beacon. 92 of them resulted in antibodies with a fair number, with enough material to actually test. And then from that, we identified 62 that were specific, and there was a strong correlation between protein binding and the FM1 cell binding, which we showed here. And then we went through, this just gives you a snapshot of what we can do on the Cartera, which again is another amazing system that we use for high throughput SPR, which is again a many-to-one type of system where you can lay down hundreds of antibodies on a chip, on the SPR chip, and then flow sequential targets over that surface to look at kinetics against those specific targets. So here is shown here is just an example of where we used anti-FC capture and then captured either antibody from crude soup or a purified antibody on each of these spots, and then we can vary the amount of antigen to get titration curve and kinetics for each of those specific antibodies. And here shown here is a snapshot of all that data. So you can see that in many cases we're getting really high affinity, really tight binding derived from the hyperimmune mouse, which is again fantastic. So you want high affinity, and the Cartera allows you to rapidly assess whether you have really high affinity or potentially weaker affinity, or maybe even no binding. So that's a nice rapid way to assess affinity. And then I'm just going to focus in here on this one specific panel, again, showing, very high affinities, maybe in this many cases like sub-nanomolar or picomolar binding, really, really tight. Whereas there's a couple others that don't show binding. We don't want those, so we'll get rid of those. In addition to using the Cartera, which you have to basically immobilize the antibody on the chip, we can also have other ways to do we can also do BLI on our octet systems as well from Sartorius. And so that allows us to not only do assays where we mobilize the target on the chip, but we also can, for the BLI system, actually mobilize the target. And in many cases, that's actually preferred, potentially specifically for multimeric targets or high or weak affinity antibodies. We find that the BLI is really nice, and oftentimes these two systems can work really nicely in concert with one another to double check affinities potentially, or in many cases one works better than the other in terms of actually obtaining the specific affinity. And then again, the Cartera system really is ideal for monomeric targets and also high affinity antibody targets. And then with both systems, we can then do epitope binning. So this is just an example of an epitope bin that probably came off of the Cartera. So you can really see that we have, from this one particular case study, multiple epitope bins against the target. So really going back to what we talked about before, kind of the hallmark of the hyperimmune mouse is the broader antibody repertoire. It allows you to then have multiple epitope bins. And as we all know, multiple epitope bins is what we want, because that will potentially leads us to more sites that have functional activity. And what we want to do in the end is actually ascribe specific functional activity to each of these bins. And then from a structural view, actually know like which ones are the key bins that we want to go after. And so we may even be able to go back later and use this information to see if we can pull out even more antibodies that hit those specific bins, so that we can get more antibodies to those functional sites on the target. Here's just more examples of how we could use unpurified antibodies to do high throughput characterization. I find that people that can do high throughput characterization using these systems using unpurified antibodies really know what they're doing, because it's really difficult to do unpurified antibody characterization, because again, you have to go through that capture step, and you actually also have to make sure you have enough antibody in your expression to actually have sufficient signal, and the Boston team does it routinely. So here's an example of a company we work with where they were using their AI ML platform for antibody discovery. We expressed over 192 antibodies in crude soup. We did binding kinetics against 4 different antigens, as well as binding to specific domains, and then did epitope binning and blocking using the assays I just talked about. And of course, like all of our customers, they were on a really tight timeline. We use the Cartera as well as the octet. And so for the kinetics, domain binding and epitope binding, which were all probably monomeric targets. We, of course, used the Cartera. And then for the ligand blocking, we used the octet. It's just an example of some of the Cartera data, looking at binding to all 4 antigens, plus an irrelevant protein. And then also this is a nice pictogram of the different affinity ranges that we're able to see. Again, these are antibodies derived from the AI ML platform, their platform, not necessarily from our mouse. And as you can see, they saw a broad range of affinities down into the sub-nanomolar range, which is great. And then we also did epitope binding. You can see that, we got several different smaller bins, as well as a really big bin down here. And then here's another graphical representation of this as well. And of course, all the antibodies that bound to domain 1 all went into the same bin, which makes sense in terms of localization to a specific domain, of course, having a similar epitope bin. And now I'm going to end with the developability assays that I've been hinting about throughout the whole talk, which is really very extensive. I mean, even in the South San Francisco site, we only have a small number of these assays relative to the suite of assays that the Boston site has. So really goes to show the time and effort that the group put in to develop these assays, which are not easy to do. So the first one is cross-interaction chromatography. So you can see how, I'm not super familiar with this assay, but I think it's about how proteins might interact with each other. Of course, HIC is a great assay for assessing hydrophobic interaction. And again, longer retention times on this assay oftentimes are indicative of hydrophobic patches that might lead to aggregation. Of course, we can do size exclusion as well to assess aggregation. We also can do affinity capture UC SINs as well, which again is you're immobilizing your antibody on a gold nanoparticle and looking for interaction of those particles. So again, that gives you an assessment of self-association of your antibody. BVP ELISA is a great assay to use for polyreactivity. So does antibodies show just non-specific binding to irrelevant antigens? We use the BVP ELISA, which was again started up by Genentech. It was a great assay for assessing irrelevant binding, which again is oftentimes indicative of poor PK. And then finally, thermal stability using our nano DSF. And then South San Francisco site, we also use the Unchained Uncle system. That gives us a great way to assess both TM as well as TAG for the antibody as well. So just to conclude, the in vivo side of Twist Biopharma Solutions has done over 100 successful antibody campaigns against a huge number of cell surface targets. We have 4 antibodies discovered by Twist, 4+. There might be a couple others that we don't know about that have been advanced to clinical trials. It takes about 29 days from immunization start to antibody sequence for a single B cell campaign, and 10 weeks from immunization start to antibody sequence for a hybridomic campaign. So again, I can't say enough great things about the Boston team in terms of their ability to discover antibodies from in vivo sources, both mice, as well as alpacas for VHH antibodies. So please contact us at this email address, as well as I'm sure Chrissy will post a bunch of other connections and links, and also follow up with you later if you're interested in a project with us. But again, as you can see from the data I showed today, we get a lot of potential sequences to move ahead. So a lot of potential shots on goal to help you be successful. And now I'd be happy to answer any questions.
Thank you so much, Aaron. That was fantastic. We do have one question so far in the chat. The question is, can you explain more about your functional assays? how do you measure antibody internalization, since there could be many different receptors that internalize antibodies in different ways? Apoptosis.
I'm just learning about this myself. If I had to guess, I would think it probably was, they probably use a secondary that has a fluorescent probe that's pH sensitive. So you express your mouse antibody, for example, it binds the target in the pen, and then you tag it with a secondary antibody that's maybe labeled with like a pH photo dye that's pH sensitive, and so that can assess whether it gets internalized and it gives a fluorescent signal, which can be read out on the beacon. So that's what I would guess is probably the assay they're using. I'm still, again, as I mentioned, kind of learning this as well, but I think that's probably how they do it.
Awesome. And we can definitely follow up with any additional questions that anyone has. Looks like that is it for now. I just want to say thank you to everyone so much for joining. Oh, one more question that just came in. What is done to improve DiversimAb's immune diversity?
There were some genetic alterations that were done to the mouse that allow it to have this more open immune system. I can't really comment specifically on what those are, but that was done early on in the formation of the company Abveris. That was really important to the genesis of the mouse.
Perfect. All right. So like I was saying before, we will follow up with any additional questions you guys may have. Aaron, thank you so much for your time today. And thank you everyone for joining us.
Chris, there's one more.
One more question. Here we go. Are you looking into discovering antibodies to intercellular targets or just surface?
Well, we can do intracellular targets. Intracellular targets are super easy too. So again, for those, you could probably oftentimes make them as soluble recombinant proteins. And so we can just do a straight soluble recombinant protein immunization. And then for the screen on the beacon, I didn't really talk about that too much, but we can, again, immobilize the recombinant protein on a bead. And then that allows us to kind of mimic the cell workflow I talked about before to look at binding of the antibodies to protein-bound beads on the beacon. Great question.
Excellent. All right.
Okay, great. Well, I really enjoyed presenting to everybody. I hope you learned a lot. I certainly did too. So, and I look forward to seeing everybody at meetings coming up. And again, if you have any questions or want to talk to any of our sales folks about a project, please follow up with us. We would love to work with you. And as I mentioned, we have great expertise in this area to make sure that you're successful in the end, because that's where we get most pleasure out of is seeing the success of our customers. Thank you so much.